In this video, I'm going to review about the chemical structure of carbohydrate. There will be 9 critical points we will be learning here. 1. Why do we need to learn about carbohydrate anyway? 2. So what is carbohydrate? 3. How can we classify carbohydrate based on degree of polymerization? 4. What are the nomenclature of monosaccharides? 5. What are reducing sugar? 6. Can you mention several types of simple sugars that are important in food biochemistry? 7. What is glycosidic linkage? 8. Can you classify polysaccharides? And 9. Can you provide some important example of glucans? Okay, let's go. Carbohydrate is an important macromolecule to be discussed in biochemistry. In human, carbohydrate play a role for energy storage and transmission. Some types of carbohydrate also possess functional activity and provide health benefit beyond nutrition. In many foods, carbs are commonly the primary source of calories, and many carbs also play a role in the food sensory characters and texture modifier. Okay, next. Carbohydrate can be classified based on their availability to be digested and metabolized in human cells. Available carbohydrates are those that are able to be digested, absorbed, and or metabolized as source of nutrient in our body. These include glucose, fructose, dextrin, sucrose, and starch. We will be discussing more about this in the next lesson, especially on how carbohydrate is metabolized in the body to provide energy or being stored as glycogen. Recently, unavailable carbohydrates also gaining more interest, though it is not able to be properly digested, absorbed, and or metabolized. This is due to its functional activities such as fiber that may contribute to stool mass, aiding digestion, and act as fermentation media for microbial gut. Okay, next. Carbohydrate reaction during food processing are very important in the development of wanted and unwanted textures and sometimes flavor too. Some of the well-known reactions include starch gelatinization and the following synerases or retrogradation, as well as non-enzymatic browning such as sugar caramelization and sugar-protein interaction in Maillard reaction. Gelatinization process is usually achieved by soaking starch in excessive water and applying heat on the mixtures. Starch granules is then absorbing water and the starch structures open up and rearrange its structure into network that entrap water. According to the starch, the ability to entrap and hold water is varied according to starch structure and content. Tubers and grain are usually contain amylose and amylopectin in various compositions, causing them to have different gelling properties. Several red seaweed extracts are known for its content of the combination between agarose and agaropectin that are able to create a very strong network upon gelatinization process and hold water for more than 1,000 times of its own weight. When the gel produced from gelatinization is cooled down, the starch may rearrange themselves in ordered structure, causing shrinkage of the starch granules. This process is referred as retrogradation. Retrogradation may expel the water out from the gel, causing both a more rigid structure of the gel and water layer outside the starch granule that we refer as syneresis or the crying gel. You might observe a layer of water in top of the agar that you store in the refrigerator. Well, this is one example of syneresis. Retrogradation and syneresis also explain why when you store your rice or bread in the refrigerator, it will harden. The gel will go into retrogradation and syneresis, and the water released from the gel evaporate into the freezer. It is more preferable if you store your leftover rice and bread in a freezer. Except if you want to make nasi goreng, it's good to keep it on the refrigerator. Well, the quicker the freezing, the least chance retrogradation will occur, and the water will freeze inside the starch granules, keeping them moist when thawing your bread and rice for later consumption. Simpler sugars such as mono and disaccharides contents are important in the color and flavor formation upon heating process, such as in the caramelization and Maillard reactions. Both reactions favor low water content and high head temperature to happen. In caramelizations, simple sugars receive heat to a point that causes them to oxidize and break apart into smaller flavor molecules, such as the acetyl that you might know for its caramel aroma. The oxidation of simple sugar also causes them to combine with one another to make color compounds such as caramellan, molecules that aggregate into small brown molecules. Okay, next. So what is carbohydrate anyway? By its chemical structure, carbohydrate can be defined as the polyhydroxyaldehyde or polyhydroxyketones and their polymer. Included in this definition are sugar alcohol, sugar acids, or sugar structure which have carboxyl group, deoxy sugar, amino sugar, phosphorus sugar, sulfur sugar, and ribose. Carbohydrate can also be classified according to degree of polymerization. Smallest entity of carbohydrate in which hydrolysis can no longer be conducted is referred as monosaccharides. Two monosaccharides join in glycosidic linkage forming disaccharides, and more is joining as oligo and furthermore polysaccharides. Okay, next. As previously mentioned, structure of carbohydrates start with simple monosaccharides. Monosaccharide is monomer that cannot be hydrolyzed into smaller product. You can say they are fundamental or simplest structure of carb polymer. Monosaccharides consist of several hydroxy groups that we refer as polyhydroxy and a carbonyl group in the middle of the carbon chain or the ketone group, or in the end of the carbon group or the aldehyde group. Monosaccharides are often available in its cyclic form, often known as Hayward projections. Monosaccharide cyclic formation from its Fischer projection is discussed in another lecture. The UPAC nomenclature for polyhydroxyaldehyde begins with aldo and ends with oze. For example, polyhydroxyaldehyde with 3 carbon atoms is called aldotriose, and those with 6 carbon atoms is called aldohexose. Glucose is an example of aldohexose, while polyhydroxyketone ends with ulose. For example, polyhydroxyketone with 4 carbon atoms is called tetrulose, and those with 5 carbon atoms is pentulose. Fructose is an example of pentulose. One of the key distinguishing features of monosaccharide is its reducing capacity. The reactive anomeric carbon, in which carbonyl compound is located, is free to react with many other compounds. That is why all monosaccharides are reducing sugars. Okay, next. When two monosaccharides join each other with glycosidic linkage, they are called disaccharides. The glycosidic linkage is formed through condensation or dehydration reaction of the two monosaccharides. Disaccharides are polar compounds that are soluble in the water. Several popular disaccharides include sucrose, the table sugar, lactose, the milk sugar, and maltose, the malt sugar. In the formation of sucrose, the anomeric carbon of both glucose and fructose went through dehydration reaction and create an alpha-1,2 glycosidic bonds. Therefore, the resulted sucrose no longer has free anomeric carbon and its reducing capacity. This is why sucrose is a non-reducing sugar. Just like sucrose, lactose is an example of heterogeneous disaccharides, as this is a result of two different monosaccharide condensation reactions. Lactose came from glucose and galactose that are connected through beta-1,4 glycosidic bonds. Digestion of lactose requires lactase enzyme, a class of beta-glucosidase enzyme that produce in the liver later in life and in relatively smaller amount than alpha-glucosidase enzyme. This is one of the reasons why fresh cow milk that have much higher content of lactose compared to breast milk should not be introduced to smaller infants, preferably under 2 years old. Well, formula is good too. Maltose is an example of homogeneous disaccharides resulted from condensation reactions of two glucose units that are connected through alpha-1,4 glycosidic linkage. Maltose also comes from the digestion starch containing amylose and amylopectin, the polymer chain of glucose. Both lactose and maltose still have reactive anomeric sugar, and therefore they are both reducing sugars. Okay, this is the first part of the carbohydrate. The next part we will be discussing about polysaccharides. See you later.